That's recorded.
Yep. Okay. Second part of CML. It's okay if I sit down, right? We don't.
Yeah.
Okay, cool. Thanks for making it, and let's get started. Well, let me just see if I'm not muted. Okay, I'm not muted. Okay, so CML is known as one of the first malignant diseases, or the first, associated with cytogenic abnormalities. So to tackle this a little bit, we need to talk a little bit about what are cytogenetics and why was this the first one. Also, the projector quality is kind of lackluster, but it's okay. Okay, that's annoying. So why was CML the first one? So the translocation event, the 922 translocation event, was observed in clinical settings, and it led to the discovery of the Philadelphia chromosome in 1960 in the city of Augusta, Philadelphia. And it's very uncommon for you to be able to trace back cancer pathogenesis to one specific mutation. Um, Leukemia specifically don't cause tumor, so it's something that you can't really visibly see, unlike other types of carcinomas that you might see elsewhere. If we're talking about benign tumors, then it stays like that. If we're talking about malignant, they go around the body. Leukemias are in plasmatic form, therefore they cannot form those clumps because the plasma of the cells has to be yes.
Is the only way to kind of diagnose leukemias and lymphomas through like platelet counts?
There are visual signs, like in the skin. In the skin, you get to see like visual, like how do I say it? Yeah, like they're like dark. They're not melanomas technically, because melanomas are its own separate thing, but you can see lesions on the skin because of the higher concentration of like
Yeah.
platelets and red blood cells, white blood cells. But it's very uncommon for you to be able to trace those back to like one specific thing that starts it. Which is why CML is like the first one. Want to add anything?
No, I guess what helps is that it's a cancer of the blood cells, right? So the fact that you have such an abundant source of cells to like diagnose from also helps. Like John said, it's not like a clump sitting somewhere, it's just all throughout your body. So I guess that helped them in their diagnosis and actually finding that, something like this exists.
Yeah, and we'll look at I mean, we've all seen visual representations of the chromosome. You get a shortened chromosome 22. And when you're studying karyotypes, you're able to see that visually too, which is how they came to the conclusion. They were diagnosing these patients, they were looking at karyotypes, and they were looking that chromosomes 9 and 22 looked different than what they normally would. So, cytogenetics. So this is the study of chromosomal properties, structure, and things of such. So here's where we analyze chromosomes we analyze DNA at the chromosomal level. And it differs from your standard analysis of nucleotidal mutations, aberrations, and things like such. For chromosomes specifically, like we were talking about last review, and like we read in the papers, there are 4 types of mutations. So these are duplications, additions, and deletions are considered in the same kind. You have inversions and you have translocations. And these are modifications that have to occur once the cell, or not modifications necessarily, they can also be mutations, that happen once the cell has already mobilized for M phase in the cell cycle. John, why does this only happen once M phase starts? This is because before M phase, your chromosomes are not going to be condensed. So they won't be in hetero.
Right.
Yeah.
Yeah.
Yeah, they won't be in heterochromatin structure for ease of access to the genes unless you are condensing for cytokinesis, which is the last step of metaphase. I mean of M phase. So visually you're able to see this in between anaphase and metaphase or metaphase and anaphase. And this is because it's once growth is done, replication is done, and you'll have the chromosomes forming. Anything you want to add?
I think you're gonna touch on this in the next slide, but translocations are unique from those other ones in that you can have a case where you don't add or lose any genetic information.
Yes.
That's balance translocation events.
Yeah. So good. You can.
So this is like I said, this is an event where you don't lose any genetic information. So whatever, switch did happen, it's all still there and just in, the wrong place compared to the wild type. And this is interesting because I don't know if any other mutations can really do this or to the extent that this does, but you can potentially, like in CML, have cases where you're asymptomatic. There's not something so wrong that your cell is showing obvious signs of just immediate death because there's like a chromosomal abnormality. It's just a simple switch and all your DNA is still there, so the cell can act asymptomatically for at least, a certain time until, other mutations accumulate or you have enough of those deformed cells where you can't be asymptomatic. Anything else?
And this is just an example. You don't have to like memorize this, but that's a Robertsonia translocation. And you have one chromosome that will be half the genetic material of the original chromosome pair, one of the other kind, and then you'll have one continuous, like longer chromosome. That's just a type of non-balanced translocation event. Yes, Jen.
So is the reason why this is bad, like, you're not losing anything, really, so is the only reason why it's bad is just when your body wants to go express a certain gene and it goes to express, it's like expressing the wrong thing?
Well, it's not like CML, like this for CML. It's bad because it happens in like a perfect storm kind of way where you get that BCR-ABL fusion. And ABL is a, cell proliferation and cell growth, like, promoter, and that's what it does, but it's so tightly regulated in the wild type. Except here, you lose all that regulation because you have that BCR addition. And so now you just have like uncontrolled growth, which, down the line, we'll talk about this in a sec, but when you're in chronic phase, it's like, okay, this exists here, but we could be asymptomatic. But once you start letting that accumulate and like play out, then you gain other mutations where, you're resistant to treatment, you have other things going on in the cell that are maybe more deathly than like the CML downstream things. And so yeah, it's bad because you just have an uncontrolled ABL now.
Yeah, and translocation events happen, like Laura was talking about, specifically in blood cells. The DNA is more because of the need for a less regulated, more random generation of a sequence of nucleotides, like we were talking about, generation of antigen binding domains and things of such for innate and adaptive immunity. The genes that are in place for like these chromosomes in blood cells specifically will have a larger malleability than, let's say, your cells elsewhere. And so that is believed to be one of the reasons why this happens specifically and causes leukemia. Okay, so diagnosis. I know we covered this in more in depth last review. But so we'll talk about two main methods. I know the problem set asks you for more, but these are the two most common ones. Damn, that's kind of counterintuitive. It's annoying me. Anyways, so complete blood counts or full blood counts, whatever you want to call them, they're the same thing. These are just routine checkup exams that people do literally for their physical sometimes. Most CML cases are caught without expectation through complete blood count. We'll talk a little bit about chronic phase specifically, but this happens without expectation because patients, like Aaron was talking about, remain asymptomatic. So they have no idea this is going on. They're going in for a routine checkup to see what they need to do. And all of a sudden they have extremely elevated white blood cell and platelet counts. Normal white blood cell count is around like less than 10,000. Here you have it sometimes reaching 200,000 plus. So that's a very, very large increase. And platelet counts, which are usually in the low 3,000s, will usually more than triple. So these are very, very high numbers. And we'll go over in a little bit to talk about why, other than overproliferation, this is not a good thing.
So I guess you can consider this more of a second line diagnosis, or maybe even third, but definitely not. I wouldn't say this is primary. Just because, you go from your CBC and, you have this abnormality in your white blood cell count, and then you realize, oh, something's up, let's figure out what this is. That could mean, there's a lot of diseases that have, those kind of symptoms. So you use fluorescence in situ pronunciation, hybridization. So this process basically involves, you have a probe, mRNA, I believe, right? So the probe is mRNA.
No, the probe, it's his own thing.
Yeah, but what is it?
There's a sequence of targeted complementary mRNA.
Yeah.
And the probe gets attached to that.
So the probe is attached to that complementary mRNA. You use a mild detergent to permeabilize the cell so that the probe can enter the cell. Probe, then you can denature the DNA so that it opens up, or you can use some sort of helicase system. Either or, there's like difference.
Yeah, there's
Either-
different
It's
methodologies.
not that important, but basically that you can open up the DNA, and if there is a match to that complementary sequence, the mRNA will attach and and that tag will be present. When you go ahead and view it using fluorescent microscopy.
The different tags will have different responses to varying wavelengths.
Yeah.
Which is how you can have different colors. So I know with this projector, it's kind of hard to see, but the green and red are BCR and ABL. I don't know in which order, and the yellow ones are the representation of BCR ABL. And Laura was talking about how you're able to tell that, to determine. And basically you need a sequence of amino acids that is complementary to both the BCR and the ABL genes, with a medium part that goes, how did she explain this?
For what the-
You have a terminal mRNA on either side that continues on, and they have to match both sequences in order for it to identify where the gene is.
Yeah.
So that you can actually tell that they're next to each other. And that's how they get the yellow probe over there.
Are the tags that cause colors like GFP based, is that why they look like?
No, the GFP is a different protein. The probes that they use aren't like proteins, they're just like, I don't really know the biomedical engineering behind it. But the probes respond differently to varying wavelengths. So when you shine UV at it, it responds with like the varying wavelength of whatever visible color it is. I don't know the wavelength spectrum enough to tell you what the range between like visible light is, but it's within the human eye's range, or the microscope's range, not the human eye. Oh. Okay. So combining treatment, that's also one of the questions in the problem set. The application of tyrosine kinase inhibitors could be used along with other chemotherapy drugs. So I wanted to, raise this question to you guys at the review to have a little bit of interaction. What do you think some examples of this would be?
I think one of the ones I talked about in the article was like GNF2, it was something like GNF2. Yeah, and they combined it with like a imatinib or one of the second gen. And it worked better.
You missed 285 TA? I have one in mind.
Chemo?
Yeah.
I don't know.
Taxol.
Oh.
What's Taxol do, guys?
It stabilizes.
So it prevents hybridization of the microtubules. So let's say that this cell that you are applying the Taxol to is about to do cytokinesis or about to pull the chromosomes apart. If you stabilize it with Taxol, it will drive it into apoptosis, and not only you're inhibiting kinase activity, you are also preventing it from moving along in the cell cycle. That's just one example. There are more examples if you want to look into like cytocholine and other chemotherapy drugs, they would also perform similar microtubule stabilizing functions or whatever is that the chemotherapy functionality is. And if you can combine that with the inhibition of kinase activity, then you have a good combination of drugs to prevent proliferation of the cell. However, this doesn't fully eradicate the problem of dormant cell lines that can just activate later on. So it is a temporary fix, and unfortunately, patients who are taking Gleevec/imatinib need to continue on taking. At least 50% of them do, as per the data for the paper in 2021, or else they go back into relapse, unfortunately. Good
Yeah.
questions? Okay. So, Gleevec was so successful. Why did our other therapies not follow suit? So, the translocation event is very, very specific. As we were talking about, once you get rid of the cap domain, the original myristoylation site, then you get rid of all the intrinsic autoinhibitory mechanisms for ABL. So what does this happen? I mean, why does this happen? This happens because you were translocating the BCR gene exactly where the original cap domain was supposed to be from ABL. And so all of a sudden, instead of having autoinhibitory mechanisms, you have a mechanism of dimerization because of the oligomerization domain. That's the coiled coil region that the paper talks about in BCR. And Laura was talking about Gleevec in class and how it was very revolutionary by the time that I was being born, 2002. So that's like 20 something years ago, 22 years ago. But because of the specificity of Gleevec to what the functionality of ABL is, that works extremely well. However, if we're talking about other malignancies that happen literally anywhere else because of any other reason other than ABL being overactive, we do not have as much control over the triggering of the event as we do for ABL. Why do I say the trigger in the event? Like Aaron was talking about earlier, this happens, but over time, mutation accruement is the main cause of why CML continues to be so overproliferative. Okay? It's not that BCR ABL isn't causing this, but it's that once you are in cell colonies that have been there for generations, mutation accruement is higher because the more you divide, the more DNA replication happens. The more DNA replication happens, the more mutations you can get. And so exponentially over time, you go from 1 to 2 to 4 to 8 to 16 to 32, 64, 258, whatever. All of a sudden, you have a bajillion new cells, each that can have a different mutation. And these happen extremely fast. And so we are not able to trace it back to one specific thing, such as BCR ABL for other cancer lines, for other causative reasons. And so TKIs only happen to work for kinases that are causative of one specific type of cancer. That's why Gleevec also works for GIST, because it works with, I'm not gonna remember the kinase name, but there's a kinase. If you're curious about it, I put it on the other PDF.
Yeah.
Any questions about that? Does it make sense? Yeah, okay.
I can add, because I just remember what I wrote about last year.
Go ahead.
I remember saying and reading about, so imatinib works and it was specialized, to work because it has like a really good affinity to the inactive form of the fusion gene, but that might not be the case for other TKIs. Like there might be less, what's the word, uniqueness in their inactive form or the kinase itself, so that, it's harder to find a drug that has like very good specificity to that. You might end up with a drug, that might bind to it, but also binds to 7 other things, which makes it super toxic. So that's why other therapies might not, be as effective. Yeah.
You look confused.
No, I'm good.
Good?
Yeah,
Sure?
She's
Yeah.
absorbing.
I'm just tired. No, I'm good.
Aaron, you're
World.
confusing people.
Dude, I'm just, I'm
You
remembering.
can tell them.
I'm
Yeah,
good.
tell
I'm
me.
actually good.
Tell me you're confused.
Are you good,
I'm
Carla?
good.
Good? Okay.
Alright.
Alrighty. 3 phases CML. Go ahead. Oh, I wanted to get this visual up so we can reference it back. Like I was talking to you guys about last time, there are more than just the myelogenous blast or the myeloblast line of myelogenous cells. We're gonna focus on just that one, but this is like a visual representation of most of the cell lines. There are more cell lines too that this visual doesn't account for, but we'll not go there.
We're lucky that this is all innate immune system, which is generally easier, and then like adaptive, which we don't care about, but it's way more complex, and we're lucky.
Okay,
Oh, okay, chronic phase. So this is your initial phase of CML. And very important, this is where imatinib is most effective. So 90% of individuals are diagnosed in this stage. So why imatinib is most effective here is because cancers get harder to treat when they accumulate more mutations, right? Resistance to drugs happens when you allow cells to just divide and divide and divide, and then through selection, you just gain resistance. So getting it early, when you have, I don't want to say like fresh cells, but younger cell lines that haven't mutated as much is going to be your best bet. This can last up to several years, and it can be pretty asymptomatic, like we've already discussed. So people can have this for up to 5 to 6 years, and not see a single symptom up until they hit their next accelerated phase. What else? So basically, you have your occasional presence of abnormal myeloblasts and granulocytes, and it's generally the rule is you have less than 10% of blasts in the blood, in the bone marrow. Am I missing anything?
and once symptoms do start to be developed, it's your typical weight loss, fatigue, fever, that most cancer patients have as a general rule of thumb for symptoms. So before I go into accelerated phase, I just wanted to go back at the visual. As we can see here, the more specific cells that we talk about, so these are the granulocytes, those 3 down here, those are the white blood cells, myeloblasts, red blood cells right here, all come from the hematopoietic stem cell, which then goes into the myelogenous stem cell or common myeloid progenitor, whatever you want to call it, there's no difference. As you can see here, besides the red blood cells, which are very much carrying oxygen up and down, all of the other ones get more specific with time, and or the more mature they are, the more specific their functionality is. And the more functional / the more mature they are, the smaller the cell gets because their functionality is so applied to like very specific things. As you can see back here, blasts are a little bigger. So, accelerated phase, it's the intermediate phase of CML. At this stage, 10 to 19% of cells that you're going to find in the bone marrow are going to be blasts. And I should have switched the order of these, but a couple of the symptoms that get added are splenomegaly, which is enlarged spleens, bone marrow expansion, and bone marrow pain or bone pain. Other than the regular fatigue, fever, weight loss, lack of appetite, these 3 start becoming prominent. And you might ask me, John, why does this happen? So splenomegaly is going to happen because you're going to start accumulating so many blood cells. You're going to have an overproduction of the initial blood cells, and you're going to start running out of space in the bone marrow, and your body is going to realize that. So then what's going to happen is these cells are going to be deposited into the spleen. And so once these cells are deposited into the spleen, they start causing the spleen to increase in size. Spleen also is not used to that elevated number of cells inside of it. And so it has to adapt to it. So you'll see some 15 to 20 centimeter big or large spleens, and that's like massive. And so with that, bone marrow expansion also happens. Your bones cannot handle the number of cells that are going to be there. This happens because mutated cells will start over proliferating, and then the bone marrow will have to expand to accommodate for all of these different cells. What's the ultimate factor into this? Once expansion happens, the patient will start exhibiting bone pain because the bone is getting fuller with cells that were not there initially. And it's in this stage that you're going to start seeing resistance to the first generation inhibitor of imatinib. And this is where the use of second generation tyrosine kinase inhibitors starts. You have plenty of second generation inhibitors in the market, the dasatinib, nilotinib, you name it. They are second generation inhibitors because they start targeting mutated cell lines that have mutated ATP binding sites where Gleevec normally acts. A common mutation that we talk about in class is the threonine 315 isoleucine mutation. I know I covered this in the last review session, but I wanted to ask you guys again, why do you think that mutation is so notorious, and why does it lead to such devastating effects?
Like signal
But why specifically because of Gleevec?
resistance to Gleevec
Yes.
because it accumulates mutations, so now it targets just the BCR-ABL, but other mutations come up.
I'm gonna draw this. I appreciate the answer, but so you're gonna have threonine, which is a polar amino acid. I'm not gonna draw the NC. We don't need. I'm just gonna draw the R groups. So threonine is weird because it looks like, so that's a CH. That's a CH2, or no, that's a CH because it goes up. This is gonna be an OH. This is gonna be a CH3, and that is threonine. Right? Or just T. You're gonna mutate that, which is a polar hydroxyl-containing amino acid, to an isoleucine. What's that gonna do? You're gonna change the polarity of the amino acid. You're no longer going to have your non-covalent, non-polar interactions happening, which is what leads Gleevec to be able to bind. And so pharmaceutical companies started targeting different cell lines that have mutated amino acids at specific spots. 3315I is just one example. I named a couple other examples that you don't need to know on the other review if you want to go look at the slides. But if you're changing the polarity, all of a sudden a molecule such as Gleevec that depends on the molecular interactions that are nonpolar will no longer be able to bind because it needs to bind specifically where it binds to. And so that is why the development of second generation inhibitors started. And for the recording, I just showed a drawing of isoleucine and threonine on the board. Absolutely. Oh, I'm glad you asked me that because I was gonna just scoot right over it. Okay, so extramedullary hematopoiesis. So hematopoiesis is this, right? For most of the time, where is this happening besides for B lymphocyte maturation in the bone marrow? Because B and T lymphocyte maturation happens in the thymus, but we don't need to talk about it. This is happening in the bone marrow, right? Inside your bones, and it goes directly into your blood vessels. Now, remember I talked to you guys about the spleen being enlarged because of the number of cells that are being deposited in there. All of a sudden, you're gonna have so many young cells in there that they're gonna start proliferating in there, causing hematopoiesis or blood generation in the spleen. And because this is not happening in the bone marrow or the medulla, it's called extramedullary because it's not in the bone marrow. So you're having so many of these younger, larger cells that you are increasing your spleen in size, and inside of the spleen, something that is supposed to happen strictly in the bone marrow, that is hematopoiesis, will be happening inside of it. So that's why it's extramedullary.
Would you say that extramedullary hematopoiesis is one of the symptoms of cholestasis, or would you just say it's something that characterizes?
It's a sign. The symptom is the stomach pain because of the enlarged spleen.
Consequence.
Yeah. Oh yeah, and the progression for accelerated phase, as the name might suggest, is much faster than your years long one for chronic phase. Any questions? Thank you for the question, by the way. No? Okay. So then we'll move along to blast crisis. So blast crisis is the most advanced phase of CML. And unfortunately, it has a very, very fatal prognosis. Mortality rates are very high when patients get to these phases. So for here, greater than 20% of your cells will be blast cells. And because mutations are getting so common in the cell lines, these cells are going to become unable to differentiate. So you might ask me, how do you get these increased blast cells? This is happening because you have myeloid progenitor cells that are duplicating, and they are able to differentiate into blast cells. But because there's so many mutations, they're no longer going to be able to differentiate into your prometacytes, into your metacytes, into your promyelocytes, myelocytes, and metamyelocytes, and they're no longer also going to be able to become platelets or granulocytes. And this is going to culminate into you having a abnormally large number of these larger blasts in the bone marrow, in the spleen, in your blood vessels. Is that a question, John?
Oh, yeah.
Okay. So extramedullary hematopoiesis continues. So this is going to happen in the bones and also in the spleen. And those three symptoms will continue to get progressively worse. So you're going to have progressively worse bone marrow expansion, you're going to have progressively worse bone pain, and you're going to have progressively worse splenomegaly. So hematopoiesis is going to continue to happen at higher rates, but the cells will not be able to differentiate into their more functional, more mature ends of the cell lines. So you're going to have decreased eosinophils, you're going to have decreased basophils, you're going to have decreased eosinophils, basophils, neutrophils. Yes, thank you. And you're also going to have lower numbers of platelets than you start with in chronic phase. But something that's interesting about this is that red blood cells, for some reason, are not affected. So you'll have a continuously normal level of red blood cells in your blood. They can be, because once you start not having enough white blood cells, and that's actually really good that you bring this up, because you start becoming anemic, you start having a lack of strength in your immune responses, your infection responses are lower, your inflammatory responses are lower, and then all of a sudden you also start developing various symptoms like the ones we just talked about of a different type of leukemia. Essentially, you go from having chronic myelogenous leukemia, which is the more advanced, more mature cells being overproliferative, to an acute myeloid leukemia, because these cells are very, very much immature, and you have a much larger than normal number for them.
Is there really nothing like currently now that exists to make it so that those cells like differentiate into their regular function?
I don't know.
I don't think so.
Yeah. And so these symptoms are very, very close to what an acute myelogenous leukemia would be, but you also see Philadelphia chromosome positive acute lymphocytic leukemia, which is when kids start developing the Philadelphia chromosome and they start having really, really bad prognosis from a very young age. So it's very sad. And I think that's it. If you guys have any questions, feel free to email us. And yeah, thank you so much and good luck.